Okay, so now let's go to a drawing activity. So this is what I do when, let me set up my iPad for this. So yeah, get out your pencils and papers if you want to follow along. But this is the way I kind of ended up memorizing and learning this by drawing it out. But I'm going to show you a way, not only am I going to draw it, I'm also going to show you my little mnemonics that I use when I was like studying this stuff and trying to draw a sarcomere and know the different types of bands, lines, and all that stuff. All right, let's see. Okay, set my iPad. Okay, here we go. Okay, so here we have our drawing activities. So what I want you to do is, if you have a red and blue pen, that's even better. And those are pretty much the only two colors you really need for this activity. So what I want you to do for the red pen, so I'm going to use red to represent actin. So red will be actin. And these will be our thin filaments. So what we're going to do, and you notice like in my cartoons, I always drew actin as like this. So I draw these jagged lines. In fact, I'm going to call them zigzag lines. And why do they call them zigzag lines? Well, the thing is that these are what you call Z lines. So that's why I call them zigzag. But that's not the official term. Don't call them zigzag lines. That's just for the mnemonic. Now, these are what actin filaments and all the nebulin, tropomyosin, troponin, these are what those thin filaments attach to. Ooh, that's a little too thick for a thin filament. Yeah, you don't have to draw all of them, but this is what the thin filaments attach to. And why are they kind of zigzaggy? Well, again, you have multiple of these in a row, so they're being pulled in two directions. So that's why they kind of look like that when I'm drawing these cartoons. It's saying that, okay, they're being pulled in two directions, that's why they have these Z lines. They're zigzagging because they're being pulled in multiple directions. And let me neaten this up a bit. So I call them Z lines, and this is, so the between Z lines, so not only are Z lines important because they anchor all the actin filaments, they are also form the border of a sarcomere. So all of this between between Z lines, between a pair of Z lines, you have a sarcomere. So that's why Z lines are important. They're the borders of a sarcomere. And in fact, that's another mnemonic I have. Now, don't spell this on I don't take any responsible if you misspell this on your lab practical, but this is why sometimes when I think of sarcomere, I sometimes say it with a Z. So I sometimes think of it as Sarcomere to drive home the point that, so again, it's not spelled with a Z, but I sometimes pronounce it in my head as a sarcomere because the Z lines form the border of a sarcomere. Okay, and then what are we going to draw here? So things that you have all those thin filaments and you also have this thick filaments. Let's draw the thick filaments a little more thick. So you have those really, really thick filaments. Now the thing is that because they're thin, these parts right here that only have thin filaments. So these are all the parts that exclude the thick filaments. So say we had another set of thick filaments here. So this part right here, where there aren't any thick filaments, this is what you call the I band. So you would have a thick fit, or you would have an I band there, but you also have an I band here as well. And I'm going to be lazy, but you would have an I band here as long as it doesn't overlap with those thick filaments on the other side. So the I band, you notice, contains only thin filaments. Only thin filaments in the I band. So when I think of the I band, I think of it as so thin, they're almost invisible. And if you look at them on that electron microscope picture, notice that these are the lighter colored areas of that electron microscope picture. So then we have, okay, so this part right here. So notice I drew this extra thick. And remember, thick filaments, these are myosin, right? So in the middle of all of the myosin filaments is the M line. So this is the M line right here. So what way I like to think of it, M line is middle of the myosin. So notice that's kind of actually in the middle, around the middle of a sarcomere as well. So I think M line in the middle. Both start with M. So that one's pretty easy. And then what we have here. So the thing is that now you have this part. Now this part, I'm going to like make some room here. Get rid of our sarcomere right here. And now you have this region that does not overlap with the, so this one only has myosin and heavy filaments, or thick filaments, I should say. So this part only has thick filaments. Notice that it does not have any of those thin filaments. So this part is what I call, or actually not what I call, this is what you call the H zone. Now this won't show on my drawing right here. So when I usually do this drawing in class, what I have everyone is turn their paper 90 degrees. So if I was to draw this, pretend I'm drawing it at a 90-degree angle, what letter am I writing here? I'm writing a bunch of H's in a row, right? And notice that this is only in the myosin part. So this isn't an official term, but I like to think of it as the heavy zone. Remember, thick, these are the thick, this is only has thick filaments. So I like to think of it as like heavy, thick filaments. And is it there's unfortunately no T zone, so I just think of like thick and heavy, or heavy being like something, a word related to thick. So this is the H zone, and this is why I also draw a bunch of H's. So I like to think of it, okay, this is a bunch of heavy, thick filaments and only, nothing else, no other thin filaments. So that's the H zone. And then we have this part right here. So now we have something called, and this is actually larger than the H zone. Let me see. Let's draw. So we have something overlapping between here and here. So this is the absolute extent of the, so this includes all of the, or actually it's the border of the thick filaments. So notice that the thick filaments extend here, but now you're including some of the actin as well. But this is a border between, or the outer limits of that thick zone, or this is the outer limits of the myosin. So what I'm showing here. But notice, so what do we have here? So this is what we call the A band. So I like to think of it as this A band right here. So the A band would be everything here. So when I think of A, I think of it as all of the myosin. And some actin. So I think that's it. All the myosin and some actin. So I think of A band, all the myosin and some actin. So A. I just keep on adding all those A terms. So that's what we have with the A band. And then I can't do that part. And then we have this part right here and this part where they overlap. Thankfully, they don't go alpha bet soup with this part, but this part I highlight right here. So where you have both actin and myosin, this is what you just called simply the zone of overlap. So thankfully they give us that one. That one's really easy. It doesn't have just some random letter. So this is how I draw that sarcomere. So again, I might re-upload this later, but again, we might run out of time if I go over it again.